Hi everyone, it's Dr. Wolfe here, and in this video, I'm going to be introducing modeling genes when it comes to cancer. So you guys just watched this video, why is cancer so hard to cure? And a big takeaway from this video is that it's really complicated in terms of how cancer develops and how we could treat it, right? Everyone's cancer is different. So in order to model this, we have to come up with a simplified model. So keep in mind, this model is accurately showing how genes work and everything, but keep in mind, it's not like this is what causes all cancers, right? It's always going to be an interesting combination of genes and how they're mutated and how they're interacting, all right? But this will give us an idea of, again, what's causing cancer. So a little bit of a throwback. Right? Let's say we have this original protein structure and there's an insertion. Remember, an insertion is going to cause a frame shift in the DNA that usually results in a completely different change of amino acids down the line. So we're going to get a deformed protein. Same thing with a deletion, just a frame shift in the other way. Right? For a substitution, if there's no amino acid change, well, then the protein's going to fold the same way. It's going to have the exact same shape. Right? And if there's one amino acid change, this is a tough one, by the way, right? You could literally have, like in the case of sickle cell anemia, just one amino acid change completely changes that protein and in effect completely changes a red blood cell. But sometimes one amino acid change really just maybe barely changes it. If you notice, the shape's just a tiny bit different than this one right here, but not too much. So it really just depends, all right? And then lastly, for a substitution, where we have an early stop codon, right? All of a sudden, you're changing all the amino acids down the line, you're going to have a deformed protein. And remember, the structure of a protein determines how it's going to function. So these deformed proteins aren't going to function well. The ones that have the same shape will. Okay. So this brings us to proto-oncogenes, oncogenes, and tumor suppressor genes. These are the genes that are going to be involved in causing cancer. So proto-oncogenes are genes that tell a cell when to appropriately divide. So when these are working, it's saying like, hey, all right, it's kind of like foot on the gas pedal. You should divide at this time, but overall, we're not usually dividing. All right. Proto is kind of like before, and onco means like the cancer. So an oncologist is a doctor that treats cancer. So an oncogene is going to be a mutated proto-oncogene that always tells cells to keep dividing. All right. So that kind of makes sense. A proto-oncogene is like before the gene's cancerous, and then an oncogene is like the gene's cancerous. All right. Tumor suppressor genes, all right, act as checkpoints that are kind of like brakes that prevent a cell from dividing until certain criteria are met. So you can think of oncogenes or proto-oncogenes as kind of a gas pedal and tumor suppressor genes as a brake. All right. So how many alleles are needed to, how many alleles need to be mutated for a proto-oncogene for the cell cycle to be disrupted? Honestly, just one. And for a tumor suppressor gene, just two. All right. Now you could get all this info. You could have gotten all this information from pages 5 and 6 of your cancer investigation packet. So this is just a warm up to kind of bring in that information. Now we're going to go a little bit deeper on why is it 1, why is it 2, why is it like a gas pedal? That's what we're going to talk about now. So you'll see the section at the bottom of page 2 of your packet, and this is just some space to write down some notes. All right. I want you to think of these genes as like this kind of strange car we have where we have 2 brakes and 2 accelerators, and maybe there's even like 2 people driving, like one person in charge of the brakes, one person involved in the accelerator. And when the car's moving, all right, especially moving fast, we can say that the cell's dividing, okay? So if our genes are functioning, all right, remember, the cell's normally not doing mitosis, it's not dividing, so we should have like both feet on the brakes. And by the way, when you're like if you haven't driven yet, whenever you're at a red light, your foot is always on the brake, all right? And that means your foot's off the gas pedal if let's say we're at a red light. And let's say our car's normally at a red light, right? Like the cell's not dividing. So what would happen if the genes are not functioning? All right, that means that there's no feet on the brakes, all right? Like literally you would have to have not just like if we have 2 brakes, not just one, but both feet off the brakes. And actually this is what happens when you're driving is you take the foot off the brake and the car actually will start to move a little bit. All right. So what would it mean to be non-functional for an accelerator? Well, that means that you're just flooring it, all right, when you shouldn't be flooring it. Like it's just like pedal to the metal type of a thing. All right. So that would be our analogy for cell division here. So let's look at the actual genes. So just as a reminder, and we could write in this space on page 2, proto-oncogenes are genes that stimulate the cell cycle in your normally functioning cells. Again, think gas pedal, but one that just like puts like the foot on the gas pedal, right, like at the right times and not too much. All right. Okay. So oncogenes, remember, are mutated proto-oncogenes. Again, think flooring it. So the oncogenes are like, we're flooring it, we're dividing, even though we don't need to. All right. So if we look here, let's say we have like our 2 chromosomes, homologous chromosomes, 2 alleles. The greens mean like functional genes. And here, if we have 2 functional genes, there's no cancer. Our foot's not flooring it. But let's say I just have one, all right, one gene that's mutated. All of a sudden, you don't need to have both feet on the accelerator. If you just have one foot on the accelerator, boom, your car's moving, all right? And then that cell, we could say, is uncontrollably dividing. So that could lead to a cancerous cell. So is this mutation dominant or recessive? Well, we just have one mutation, and that one mutation caused our cell to divide. So that's going to be a dominant trait because it was just one allele was needed, one mutated allele. All right, so let's look at tumor suppressor genes. All right, again, these are genes that slow down or stop the cell cycle. Think brake pedal. All right, and so let's say we have no mutant alleles. Clearly, we're not going to have cancer. Let's say we have one mutant allele. All right, there's still going to not be cancer. Why is that the case? Well, in our kind of strange and car analogy, you got 2 brakes. You got 1 foot, 2 feet on the brakes. One goes off, right? Then all of a sudden, not all of a sudden, you still have 1 foot on the brake, so that car's not going to move. However, if you have both feet off the brake, that car is going to start moving, right? Your cell is going to start dividing when it shouldn't be. So is this type of gene, is this like a dominant or recessive trait here? This is going to be recessive because you need to have 2 of the mutant alleles to get a different phenotype, to get a different function. So let's look at an example of this, all right? Now, this is just like a general slide. This isn't in your packet. But overall, these are going to be our rules for our models. One gain of function, or in other words, one mutation for your oncogenes or your proto-oncogenes, that's going to cause the cell to divide like a cancer cell. Or it could be maybe like everything's checking out for the like proto-oncogenes, but your tumor suppressor genes, all right, have 2 loss of function, 2 recessive mutations. That's going to cause the cell to divide like a cancer cell. So let's look at an example. Patient 1. The organism inherits the following mutation. So this is essentially what they got from their parents. To the maternal chromosome, there's an insertion found on the MYC gene. So, okay, insertion, that's going to cause a different shape of our protein. So I'm going to model that in as red, non-functional. All right. During the organism's lifetime, so this is like the mutations they're accumulating, the maternal chromosome, so here's the maternal chromosome, there's a deletion to both the P53 and the BRCA gene. So a deletion, all right, again, is going to cause a different shape in a protein. So these are going to be non-functional proteins, again, all on the maternal chromosome. And then everything else I'm just going to draw green because there's no mutations there. I'm going to assume that they're functional. So now I have to talk about, okay, would the cell divide like a cancer cell? And I'm going to talk about I'll always talk about the oncogenes or the proto-oncogenes and then the 2 tumor suppressor genes. And I always like to use this format. What did you model? What's going to happen as a result? So 1 MYC allele is non-functional. That's what I modeled right here. So it would tell the cell to divide uncontrollably. All right. What would occur in the cell with the tumor suppressor mutations? All right. So again, 1 allele of both P53 and BRCA is non-functional. All right. But just 1. So each gene does have a functional allele to prevent the cell from dividing. But remember, according to our rules, we're going to say that, hey, you just like, as long as like you have your foot on the accelerator, all right, that cell's going to start to divide uncontrollably in this case. All right. So we would say that the cell would act like a cancer cell. And the reason why we're saying it's acting like a cancer cell is because, all right, cancer kind of goes in these different stages. You can start to have these cells that are dividing a little bit uncontrollably, but your body's able to take care of it. All right, and then we'll see over time is that these cells start to accumulate more and more and more mutations, all right, where they finally become very, very cancerous. So if our cells dividing how it shouldn't be, either due to 1 oncogene or 2 mutated tumor suppressor genes, we would say that the cell is going to start to divide like it's not supposed to, like a cancer cell. All right, well, thanks for watching. You could use this to complete the rest of your pages.